The size of actinomycetes ranges from 0.5 to 1.5 micrometer. 0.5 to 1.5 micrometer. Actinomycetes population is largest in the surface layer of soils. Actinomycetes population is largest in the surface layer of soils and gradually decreases with depth. Individual actinomycete strains. Actinomycete, you don't put the S. Individual actinomycete strain are present in all soil layers. They are widely distributed in the soil with estimated values ranging from 10 raised power 4 to 10 raised power 8 per gram of soil. Ten raised power 4 to 10 raised power 8.
They are widely distributed. Estimated value ranging from
Per gram of soil. So with this, all this type of information will help when you are doing a project. Now you are to isolate from the soil actinomycetes. Then you isolated, you use 1 gram. Then your own is saying 10 raised power 1 per gram of soil. You know that something is wrong. Your own is saying 10 raised power 20 per gram of soil. You know that something is shelled here. That's why it's good. The theory is very good. Then when you get to the field, what you now get, when you now get a value that is very far from already established value, then you have to repeat that experiment. But that does not mean that if your values are different from what is established, you should not report it. If you are very sure that is what you get, because maybe discovering something. But in most cases, this thing has been tested and tested. I don't know the kind of soil you want to use, that you now get something that is quite different from some of these values. So, somebody that is doing an experiment or a project, if you know the theory very well, when you are doing the practical aspect of the project, you will know how to interpret your values. After the project, the experiment, you'll be asked to discuss. You get the results, you'll be asked to discuss. Your values is 10 raised power 2, your value is 10 raised power 3, your value is 10 raised power 4, yours is 10 raised power 5, 10 raised power 6, 7, 8. Then you discuss it. In this case, you go and you check, this is for averagely any kind of soil. So if you are looking at a particular kind of soil, you are going to get some particular range of value, not spread out like this. So somebody is working with a soil in ABU's area, the value can be 10 raised power 6, it's still within the range. Somebody is working on a soil in Uniport, the value can be 10 raised power 4 or 5, it's still within the range. But it's different from the ABU. You get it now? Somebody is working in Ilorin here, getting 10 raised power 8, it's still within the range. But it's not different from ABU, and it's different from Uniport. So you must know what happened in Uniport, you must know what happened in ABU research, and you must know the standard value. So you can now compare that this value I got is within the standard range, but it's different from what was reported in ABU, which is still within the standard range, and different from what was reported in another place. And the same thing was reported at Kwasu. You get it now? You must have similar, the same kind of result. and you must have different. When you come to discussion, many students don't they don't even know what discussion is. Until maybe after many years, and they come back and then they look, say, is it the discussion I wrote in that my project? So let's get it straight now. And then all these things are not just for exam purpose, also for experimental purpose. So if you carry out an experiment, now you can now refer back. This material information can be found in most microbiology textbooks. Just lay hand on anyone, then you can use that textbook as a source, as a reference. You must also have a reference. And avoid using personal communication. According to Dr. Adetitun, personal communication. According to Professor Sule, personal communication. What about if that time he said he said the wrong thing? So, based on that, make sure that you use text, written text, for your reference. So let's go on. They grow as hyphae like fungi. They grow as hyphae like fungi, resulting in the characteristically earth smell of freshly turned healthy soil. Earth smell. Of freshly turned soil. Streptomyces is the most abundant species of actinomycetes in soil. Streptomyces is the most abundant species of actinomycetes in soil. This is followed by other species like Micromonospora. Thermoactinomycetes and Nocardia. Almost 90% of actinomycetes genera have been isolated from the soil. Almost 90% of actinomycetes genera have been isolated from the soil, where they often form much branched hyphae when growing. Which then break up into spores. Either by the tip of the hyphae, producing one or two spores, actinomycetes in the soil are mesophilic. Actinomycetes in the soil are mesophilic organisms that are sensitive to acidity and waterlogged soil conditions. Optimum pH range is 6.5 to 8. Optimum pH is 6.5 to 8. Some positive effects of actinomycetes in soil are They decompose the more resistant and indecomposable organic substances. They decompose the more resistant and indecomposable organic substances and produce several dark black to brown pigments. Which contribute to the dark color of the soil humus. Which contribute to the dark color of the soil humus. The moment you say you don't get it, I won't repeat, but if you just keep quiet, then I can just repeat it. But the moment you say you don't get it, that shows that you need to be fast. So, which contribute to the dark color of the soil humus? Full stop. These organisms are also responsible for the subsequent decomposition of humus. These organisms are also responsible for the subsequent decomposition of humus in soil. The next point, actinomycetes are important in agricultural soils. Actinomycetes are important in agricultural soils because they contribute to the carbon cycle by fixation. How do they contribute to the carbon cycle by fixation? How do they contribute to the carbon cycle by fixation? How do they contribute to the carbon cycle by fixation? Class rep. How do they contribute to the carbon cycle by fixation? That boy in white with cap, with glasses. How does actinomycetes contribute to the carbon cycle by fixation? Yes. The lady in red.
Because they have been breakdown.
Yes. How does actinomycetes contribute to the carbon cycle by fixation? Those in petroleum microbiology, where are you now? Where are you? We talked about carbon cycle last week. So how does actinomycetes contribute to the carbon cycle by fixation? All right, let's go on. By fixation and decomposition. By fixation and decomposition. How do they contribute to the carbon cycle by fixation? How? How do they contribute to the carbon cycle by fixation? How do they contribute to the carbon cycle by fixation? How?
Because they help in decomposition.
How do they contribute to the carbon cycle by fixation?
They break down complex matter.
So when you break down complex matter, is that fixation?
Fixation.
Which element do you say something fixing something? Which element is that? So we now say carbon cycle by fixation. What's that? So what else is that? Yeah? Those who did petroleum last week, where's your hand? Raise your hand, let me see. Those in petroleum class last week. The nitrogen cycle. Okay, nitrogen cycle. Nitrogen fixation by what?
Bacteria.
By what?
Bacteria.
Nitrogen fixation from where? From where? The nitrogen that is fixed is from where?
Atmosphere.
From the atmosphere. So if carbon is for the atmosphere and is fixed, what is the process called?
Fixation.
Something you have known since primary school. You have known it since primary school. Primary. If I say it now, I say, yes. It's photosynthesis now. We're talking about carbon. Nitrogen cycle, nitrogen in the atmosphere. The bacteria in the root node will fix it directly. Nitrogen fixing bacteria in the root node. Nitrogen in the atmosphere to fix it. This one too is by they said these substances are also responsible for the subsequent decomposition. They contribute to the carbon cycle by fixation. Photosynthesis now. How does a goat contribute to the carbon cycle? A goat, how does it contribute to the carbon cycle? What? But through respiration, is that the only way? Which other way?
Decomposition.
When it's still alive, it will decompose. What? Death followed by decomposition. How does a tree contribute to the carbon cycle?
Photosynthesis.
Photosynthesis, respiration, and what?
Decomposition.
Burning of wood. Then even death followed by decomposition also. So please let's be innovative. Don't just be. This is what they taught us. So this is what I'm going to write. So think wide. And that brings me to the one matter I want to raise. Now you finish for microbiology now. You start looking for job, and this is soil microbiology, Abi. You start looking for job, and the jobs are so many in Nigeria, Abi. Everywhere there is job. So if you can think out of the box, you're going to make headway. But if you just stay in the normal, this thing, finish, look for job. I'm looking for job. There are many things to be talking about in the different courses, in the different subjects you are doing or courses you are doing. You see that there are careers inside already. There are businesses inside. When we come to sewage, fertilizer. So microbes are involved. So when you finish, people are looking for fertilizer, farmers everywhere. So why don't you go to fertilizer production? Then there's a problem of solo businesses in Nigeria. Just one person, you just finish, go and start one business. He doesn't have money to rent, he doesn't have money to do, he doesn't have connection to be able to reach many places. So why can't 5 of you in the class, as you start thinking, start sitting down, as you studying, also form a group, business group from the distance, just be discussing business idea from everything you are doing, maybe just once a week. Then maybe later on you can now take it and put it on the table, on the ground. So this one is good in marketing, this one is good in
Connections.
some other area, this one can get the finance. So one person get the finance, the other person is doing another something, the other person is doing the other something, then it runs. Within a few years, it becomes big, then you come and be employing other people that are coming. Then you can contact me to help you to recruit other students that are graduating. But if everybody is going out, looking for job, and jackpot everywhere, there will be issues. Olam, I thought Olam is Nigerian company that is where Olamide. Recently I just got to know it's owned by Indian. Somebody was presenting the IT report. It says Sam Pharmaceutical is owned by Indian again. So which one can be in Nigeria now? So you get it. So Nigerians are running away. Indians and other countries are coming here to. So let's think, it's good to jump out, but also look inward. Actinomycetes form associations with some non-leguminous plants and fix nitrogen. Actinomycetes form associations with non-leguminous plants and fix nitrogen, which is then available to both the host and plants in the near vicinity. Which is then available to both the host and plants in the near vicinity. Vicinity. Full stop. They are the leading group of soil microorganisms. They are the leading groups of soil microorganisms that play a major role in the recycling of organic matters in the environment. In the recycling of organic matter in the environment by the production of hydrolytic enzymes. By the production of hydrolytic enzymes. They possess a direct or indirect mechanism that favor plant growth. They possess direct or indirect mechanism that favor plant growth while improving the availability of nutrients and minerals. While improving the availability of nutrients and minerals, synthesized plant growth regulators and suppression of phytopathogens. They have the ability to act as biocontrol tools. They have the ability to act as biocontrol tools. Biocontrol tools. I gave you assignment last time. You didn't submit it. You didn't submit the assignment. So okay. Some negative effects of actinomycetes in soil are some products of actinomycetes might affect the microorganisms in the soil. Some products of actinomycetes affect the microorganisms in the soil. And this reduces the diversity. This reduces the microbial diversity of the soil. This reduces the microbial diversity of the soil. This reduces the microbial diversity of the soil. But you say you did IT now. And I say zone of inhibition.
Okay,
Yes. Control tools.
So everything I have to explain, I will not be able to explain the exam. But the question will be simple, Sha. The question will be simple, but I will not be able to explain the exam. It will be a very cheap question. It's antibiotics now. Don't you know that actinomycetes is a major group of antibiotic producers? I just said it. Some product produced by actinomycetes may affect microorganisms of the soil, resulting in decreased diversity. So it is those products is produced to be toxic to others, and then they are going to die off, reduce in number. So anywhere some actinomycetes are, some organisms will not be in that soil. So when you are looking at all the bacteria, all the organisms in that soil, some are going to be missing. Why? Antibiotics produced by actinomycetes. Pathogenic species of actinomycetes, continuation after full stop. That's another point. Pathogenic species of actinomycetes result in plant diseases that affect plant health and crop loss. That affect plant health and crop loss. Protozoa in soil. Protozoa are unicellular eukaryotic microorganisms that lack cell walls. They are motile heterotrophs that obtain food by ingesting bacteria, yeast, algae, small protozoa, and organic matter. They are motile heterotrophs that obtain food by ingesting bacteria, yeast, algae, small protozoa, and organic matter. Free-living soil protozoa fall into three categories. Free-living soil protozoa fall into three categories. Flagellates, amoebae, and ciliates. Flagellates, and ciliates. These single-cell animals differ in shape. These single-cell animals differ in shape, size, and distribution. Differ in shape, size, and distribution. With some protozoan species found in land habitats like soil. With some protozoan species found in land habitats like soil. Some examples of protozoa found in soils are amoeba, ciliates, and flagellates. Numerous heterotrophic flagellates. Numerous heterotrophic flagellates and amoebae are available in agricultural soils. grassland, forest soil, base sediment of fresh water, coastal and marine waters, base sediments. What does that mean? It's bottom that is there. I don't want to use that word because some of you will start thinking of another thing now. The presence of protozoa in the soil is influenced by the presence of living and dead plant roots. The presence of protozoa in the soil is influenced by the presence of living and dead plant roots and the organic content of the soil. Full stop. The inhabitation of soil by protozoa. The inhabitation of soil by protozoa depends on the structure and texture of the soil. Full stop. Mastigophorans. Mastigophorans, that is flagellates. Tend to dominate in drier soils. Drier. The soil that is dry. While celiophorans are abundant in moist soil. Full stop. Cultivated soil and clay soil are predominated by flagellates. Cultivated soil and clay soil are predominated by flagellates. Cultivated soil and clay soil are predominated by flagellates and amoebae. While the soil of coarse texture, coarse texture consists of large flagellates and ciliates. First off, some protozoa may remain in a symbiotic relationship. Some protozoans might remain in a symbiotic relationship with other microorganisms like bacteria and fungi. With other microorganisms like bacteria and fungi. What is the difference between you are turning your face away? What is the difference between symbiotic relationship and mutualistic relationship?
What's go explain that?
We have two organisms. So it's either one of them is benefiting.
Mutualistic relationship is the relationship of organisms, of two organisms. In such a way that it's either both of them are benefiting, or one of them is benefiting, or one of them is losing, which is negative. But when it comes to symbiotic, both of them are benefiting.
Thank you. Symbiotic relationship or mutualistic relationship.
Symbiotic relationship is just basically the relationship between two organisms. While mutualism is more the relationship between two benefiting mutual benefits.
Skip up now. Give back to you.
Yes, that's what I would do. There are relationships which both are independent. They are dependent. Like there must be two organisms. And that was what I would do. So both of them will benefit.
Thank you. Second person does it. If I doesn't try, didn't get it. So, don't worry, you're going to take that in. By the time the fourth lecturer comes, we're going to take you on that. Different types of symbiotic relationship. In secondary school, they say symbiosis. But now, symbiosis has a new, different definition. So the symbiotic relationship can be commensalism, it can be proto-cooperation, it can be competition. It is still symbiosis. While mutualistic means that both are benefiting. But symbiosis can also, mutualism is a subset of symbiosis. Competition is part of it, symbiosis. Commensalism, where one benefits, the other does not benefit, is part of symbiosis. Some positive effects of protozoa in soil are protozoans are crucial in terrestrial ecosystems. They are crucial in terrestrial ecosystem, where they act as bacterial consumers, leading to mineralization of organic soil nitrogen to form ammonium. Bacterial consumers, leading to mineralization of soil organic nitrogen to form ammonium. When you come to nitrogen cycle, details will be given to you. Protozoans community in the soil can also be used to assess. Protozoan community in the soil can also be used to assess and monitor the changes in the biotic and abiotic component of soil. Protozoan community in the soil can also be used to assess and monitor the changes in the biotic and abiotic component of soil, thus acting as bioindicators of the soil. Protozoa have been found to increase plant biomass independently. Protozoa have been found to increase plant biomass independently of nutrient contents in plant tissue. Independently of nutrient content in plant tissue. Full stop. Many protozoan species feed on bacteria. Many protozoan species feed on bacteria and other microorganisms. Many protozoan species feed on bacteria and other microorganisms, which enhances the nutrient cycles and the energy flow between the microorganisms, animals, and plants. Some negative effects of protozoa in soil are the presence of protozoa in soil affects bacterial diversity. The presence of protozoa in soil affects bacterial diversity. This is because most bacterial communities in the soil are the source of food for protozoa, or for protozoans. Some protozoa might be harmful to the plant, which decreases crop health and crop yield. Viruses in soil. Viruses are genetic elements that can replicate independently of a cell's chromosomes. Viruses are genetic elements that can replicate independently of a cell's chromosomes, but not independently of cells themselves. Viruses are smaller than bacteria and range in size from 20 to 30 nanomicron in diameter. Viruses are obligates. Viruses are obligates. Intracellular parasites of bacteria, fungi, insects, plants, and animals that inhabit the soil. Viruses can act as dormant structures or particles that can survive for a long period in different habitats. Viruses can be found anywhere in the world where there is life, there is hope. Anywhere in the world where there is life, as viruses are obligate parasites. As viruses are obligate parasites. The concentration of viruses in the soil has been estimated to be 10⁹ virus particles per gram dry weight. 10⁹ 10⁹ virus particles per gram dry weight. The majority of the soil viruses are tailed bacteriophages. Tailed bacteriophages.
The majority of some viruses are tailed.
That prefer wetland forest soil over drier agricultural soils. Tailed bacteriophages that prefer wetland forest soil over drier agricultural soils. Some of the common viruses inhabiting soil include small spherical virus particles, similar in size to single-stranded RNA. Similar in size to single-stranded RNA. Containing bacteriophages of the Leviviridae family. Of the Leviviridae family, or to some plant viruses, and larger spherical viruses, similar to the double-stranded DNA containing viruses. Of the Totiviridae, T-O-T-R, Viridae, D-A-E, comma, Patiti, P-A-R-T-R, Patitiviridae, and Chryso, C-H-R-O, C-H-R-Y, S-O, Viridae, Chrysoviridae families. Soils act as reservoirs of viruses. Soil act as reservoirs of viruses, but these are probably not entirely static reservoirs. But these are not entirely static reservoirs, as at least some viruses seem to move readily between environments. Positive effects of viruses in soil. This is by transferring genes between microbial hosts. This is through the process of transferring genes between microbial hosts. This is a microbial host, another microbial host. By horizontal gene transfer. By horizontal gene transfer. The gene transfer enables the transfer of beneficial characteristics between different communities. The gene transfer enables the transfer of beneficial characteristics between different communities. Another way that viruses in soils have potential benefits for plants is by infecting organisms. by infecting organisms that are pathogenic for plants. Organisms that are pathogenic for plants. So the virus will infect the organism, the what is going to infect the plant. So that organism that will infect the plant, the virus have infected it, so it cannot infect the plant. I was supervising and vigilating one exam in one university many years ago. So I observed that a particular lady was giraffing somebody. So you know what I did? Who should I move? Is it the giraffe or the person that they are giraffing? So the person, I didn't move the giraffe. I left the giraffe alone. The person giraffing, I left her alone. Because if I move the person that is giraffing somebody, will say, you have moved me. I left the person giraffing. The person they are giraffing, I just say, you go to that place. So the person that is giraffing is still there. He's now looking, what's happening now? So if possible, we would have gone to the other place and go and continue to giraffe. But various populations may also act as reservoirs of genes. Various populations may also act as reservoirs of genes involved in all the biochemical functioning. Various populations may also act as reservoirs of genes involved in all the biochemical functioning of their microbial hosts. And by recombination among themselves during co-infections could be a source of new gene variants. Could be a source of new gene variants. New gene variants. We are looking at benefits, are we?
Yes, sir.
We are looking at benefits of viruses.
Yes.
A virus can take the horizontal gene transfer, can take some particles from chromosomes. The virus will come and infect A, take the good chromosomes, the virus will go and infect B that has not too good chromosomes. We take the good chromosomes from here and put it here. That's beneficial. You get it now?
Yes, sir.
Like some students too, you come from secondary school, not too good students. The lecturer is teaching and asking you questions, making sure that you that are sitting at the back, you are answering questions, because those who are sitting at the back are not too good like that. So this lecturer go to the back, like these young people now, I brought them from the back, this place. Hey, let now, see where this white people now. See his face? And this man is not, he was coming in front. Every class is coming in front. You understand? So gradually, you not shoot horizontal gene transfer. Now become good. Then when you come out of university, they say after 400 level, now say, is this boy in secondary school? He didn't do anything now. What? In secondary school, he didn't do anything. Why is he? What happened to him? And then look at him, he's a graduate now. Then he will be saying, yes sir, well done sir, yes sir. You get it now? So not all viruses are bad. Some are also good. Viruses of different microbes in the soil. Viruses of different microbes in the soil as pathogens have an essential role in regulating the population structure of their microbial hosts. Some negative effects of viruses in soil are they act as plant pathogens. And they reach the plant through mechanical means. What do you mean by mechanical means? What do you mean by mechanical means? You have a virus that affect the plant as pathogen, and it affects the plant through mechanical means. These are plants. These are root tree. It's a nice system. What is by mechanical means? When the virus is here and it's attacking the plant through mechanical means. Regular. What is that? The one is very large. As I'm working in the one of power. So one, go and show mechanical means. That plant. Is it correct? Is it correct?
What were you saying before?
Don't worry. You know it by force. When you remember that this man will call me in the class again, let me read this before. With this now, we read the note very well, are we? But you know that if I go class now, that is Adetitun now will call me. Because I said mechanical means. So you are looking. Okay, let me change the English now. Mechanical injury. You want to change your answer? Mechanical injury. You want to change your answer? You want to change your answer? Before I say mechanical means now mechanical injury. What? You don't want to change the answer? You want to try? You want to try? You want to try?
Actually, when you say this one is not plant.
Our tradition. Once away organization by 5 Are you the one?
This virus here.
And this is going toward the plant. How will it get to the plant if the virus is not mechanically injured?
Maybe an insect that's carrying the virus from an infected plant. It's now eating the leaf of this infected plant. So why is it eating the leaf of this infected plant? So it just causes mechanical injury to this infected.
The virus will cause mechanical injury to the plant.
The vector that is carrying the virus.
What is the vector?
It can be the insect.
But that is none of your business now. Even what caused the mechanical injury is not your business now. That's not your business. I said among the virus community in soil, distinct proportions are plant pathogens. So the virus is a plant pathogen already that reach the plant via mechanical means.
Like chew insects, chew.
What? Now, let me use your
You do not need penicillin and
this guy now.
you do not need to it. I'm asking you.
What organism is normal flora of the skin?
Stars.
And it's normal and it's here now, Abi?
Yes.
As my body is on his hand, and it's on my hand. But nothing happened to me. When you now bring scissors or pin and tear this place, so what happens? The mucus on the skin will get access, will enter inside. It's because of the mechanical injury, because of the wound. If there's no wound, it's intact. The moment there's a wound, sarcophores anomalies on the skin will now enter inside the skin. The moment it enters inside the skin and goes to the blood, it will cause infection. Yes, now see through mechanical injury to the plant. Change the plant now. You now go, you want to do agbo. This mango tree, I mean. You want to do agbo, you now go, you are start cutting it down. Now take the back, you go and do your agbo. Then the virus is there, it went out and then cause infection. The same thing sometimes. All these parts is agbo is not plenty. It is the root that the agbo is plenty. You go and cut the roots. How many things you that before? You will cut it to the side and go concentration is here. You go and cut it. Remember now this stomach was so we are talking about the roots. So the virus is already in the solution. It's in the different part of the roots. And then to get there and then they have cut it. Then to enter the plant and infect the plant. The virus is a pathogen, but if there's no mechanical injury to the plant, it will not be able to access the plant. As I reach the plant via mechanical means, command nematode vectors or fungal vectors. Can you see mechanical means? That's you going to cut the plant. That's mechanical means. Then it can also be through nematode vector or fungal vectors. You can see insect vector is not there.
Yes.
Because I'm talking about soil. Now you are talking about the something that is flying. This man is talking about the wind. Which kind of wind affect that something? We're talking about soil now. So we now see question. We're talking about the leaf. I'll just be doing like this for you. We are talking about soil microbiology. So you bring it down to the roots. So those are negative effects. Think viruses may even affect the physical and chemical properties of the soil. Viruses might even affect the physical and chemical properties of soil by affecting the biotic and abiotic components of the soil. By affecting the biotic and abiotic components of the soil. The next thing we talk about is nematodes in soil. And then we also talk about cyanobacteria in soil. I will send the material for these two to you.
Thank you, sir.
By tomorrow, I want to look at another aspect